It's my great pleasure to introduce Irene Kaplow, who's I've known her for a while now. I know she's a fantastic person and a fantastic scientist. Her research has been really awesome to witness over the past few years, and she's had a really beautiful paper come out just recently, which I believe she's going to be talking about today. So I think it's going to be a really big treat for us to listen to her. But a little bit about Irene. She's currently a research scientist at Duke in Charles Gersbach's lab. And previously, she's done her, as I believe she's going to be speaking about her work at CMU, Carnegie Mellon, in the Fenning lab. But she previously got her PhD at Stanford with Anshul Kundaje. I'm sorry for his last name, and Hunter Fraser, where she's done a lot of really great work looking at DNA methylation and transcription factor binding. And then previously, before that, she did her undergraduate work at MIT. She's won a lot of different awards and postdoctoral fellowships, including the Lane Postdoctoral Fellowship. She previously had as a grad student NSF Fellowship, and she's gotten a lot of other really great accolades and awards. And I'm really excited to hear her present today. And thank you so much, Irene, for agreeing to present. And I'll leave it to you.
Thank you so much, Christine, for the extremely flattering introduction. It is an amazing honor to be on this symposium, having been inspired by many previous talks in this. And today I'm going to be telling you about relating enhancer genetic variation across mammals to complex phenotypes using machine learning. So over the last couple of days, I've been really struggling with how to begin this talk. The last few days have been really difficult for me. The work I'm going to be presenting today is not just a result of work I did in the Fenning lab, but of many failures and smaller projects I contributed to earlier in my career. And to give a proper acknowledgements for this talk would take more than the 20 minutes I have been given. So I'm going to begin by acknowledging the people who got me into the field of transcriptional regulation when I was first starting out my career. I'm then going to spend most of the talk telling you about my work, and then I'm going to conclude by acknowledging the people directly involved in this project. So when I was 21, I had never heard of nucleosomes. At MIT, I was a math major and was lucky to have the opportunity to work in Bonnie Berger's computational biology lab. And my junior year of college, both Bonnie and multiple people in the group encouraged me to broaden my intellectual horizons by spending a summer abroad. And specifically, they suggested doing an internship in the lab of Iran Segal at the Weizmann Institute of Science. And this lab specialized in transcriptional regulation, which I knew was something that happened, but I didn't know what nucleosomes or transcription factors or histone modifications or anything involved in those mechanisms were. And a wonderful postdoc, Maya Dadiani, this is her with the microscope that she spent over a year learning how to use so she could do her experiments. And troubleshooting many challenges, spent a huge amount of time teaching me about transcriptional regulatory mechanisms so that I can make valuable contributions in the lab. And the specific project we were working on was studying how promoter sequences affect gene expression. And this idea of effective DNA sequence on gene expression has been tremendously influential in my work. You're going to hear a lot more about that later in the talk. And you can learn more about the specific project in the paper we published in Genome Research. And there are a lot of things I could say right now. When I was at synagogue Saturday morning, services were interrupted and we were informed of the murder of hundreds of Israelis, from infants to elderly people. And I just wanted to say that my heart and my prayers are with the transcriptional regulation research community in Israel that has played such a fundamental role in my career. So today I'm going to be telling you about a project that stems from the incredible diversity of mammalian phenotypes. And specifically, I'm going to be focusing on phenotypes that have evolved multiple times in mammalian history, suggesting that there is some kind of shared genetic mechanism underlying their evolution. And one phenotype I'm going to telling you about is large brains relative to body size. So humans have much larger brains than cats and rats, and even than those of our closest relatives, chimpanzees. Yet dolphins, which are much further from us than chimpanzees are, also have large brains, we can plot body weight on the x-axis and brain weight in the y-axis. And we see is that generally there is a linear relationship between the two, with humans and dolphins having much larger brain weights than we'd expect based on their body weight. And while my passion is understanding transcriptional regulatory mechanisms underlying phenotypes like this, understanding these mechanisms has the potential to provide insights into human disorders, such as microcephaly, which is when a human is born with a brain that is too small. For the duration of this talk, I'm going to be just referring to this deviation of brain weight from what we'd expect based on body weight as the brain size residual. So the first thing we thought to do was to see if there are protein coding differences between species that might be associated with their evolution of brain size residual. And this was work done by Morgan Worthland, a former postdoc in the Fenning lab, and she was unable to find any protein coding regions associated with the evolution of this phenotype. What this suggests is that this phenotype has evolved at least in part through gene expression. To explain what I mean by this, let's consider a toy example of an enhancer bound by a transcription factor in humans that loops in 3 dimensions to a transcription start site to activate gene expression in the brain in a way that enables humans to have large brains. And then let's consider our closest relative, the chimpanzee, which has a smaller brain size residual. Now, a chimpanzee might have a difference in sequence at this enhancer, which leads to a difference in transcription factor binding, which leads to a difference in gene expression of the gene regulated by this transcription factor in the brain, which can lead to a difference in brain size. What this suggests is that there are multiple approaches that we can take to use genome sequences to study phenotypic diversity. First, as I mentioned, we can compare protein coding sequences, but we can also compare enhancer sequences. A previous study set out to find enhancers whose differences between species are associated with brain size evolution, and this was published in 2011, and they found a region that was lost in humans but present in the other species whose genomes was available. So I was privileged as a postdoc to be part of the Zoonomia Consortium, which sequenced and aligned the genomes of over 200 mammals. So now that we have all these genomes aligned, I decided to ask the question, is this enhancer also lost in the cetaceans, which are the whales, which have a large brain size residual? And to my surprise, I found that this enhancer is present in all of the cetaceans with genomes in Zoonomia, suggesting that the loss of this enhancer in humans is not the entire story of brain size residual evolution. What this means is that if we really want to understand how brain size has evolved across species, we need to study brain enhancer activity in a more diverse set of mammals than was available at the time of the previous study in 2011. So the first thing I thought to do was use an existing nucleotide level conservation-based approach. And using this, I found one enhancer associated with the evolution of brain size residual. So I looked at some Hi-C data to see if it interacted with the transcription start sites of any genes previously implicating brain size, and found that it did not, suggesting this enhancer is probably also not the entire brain size story. However, I think that these conservation-based approaches may be causing us to lose out on a large number of enhancers involved in phenotype resolution. And this was my hypothesis for a few reasons. First, most mammalian enhancers have tissue-specific activity, but conservation information is tissue agnostic. Second, enhancer function can be conserved when nucleotide level conservation is low. A beautiful study about this was published 2 years ago that identified an enhancer in human whose function is conserved in zebrafish and even in sponges. So the little colored boxes in this figure are transcription factor motifs. And what you can see is that the same motifs are present in human zebrafish and sponges, which enables the conservation of enhancer activity, but they have different orders in different species. So if you line these sequences up one by one, you would not see especially high nucleotide level conservation. Thus, how should we be identifying enhancers associated with phenotypes that have evolved through gene expression? As I mentioned, while nucleotide level conservation-based approaches have led to a number of exciting results, they're likely to miss out on a number of relevant enhancers. Another option is to experimentally assay enhancers in tissues involved in the phenotype. But the reality is that we cannot obtain most tissues from most mammals. So this approach is infeasible and should not be feasible, in my opinion. Thus, how should we be doing this? As a postdoc, I led the development of a research program to efficiently trace the evolution of phenotypes that have evolved through gene expression. And what I mean by efficiently is without having to sacrifice large numbers of mammals. So the way that we do this is we first identify candidate enhancers in a small number of species from which we can often obtain primary tissue data. We then use the sequences underlying enhancers and non-enhancers to train a machine learning model to predict enhancer activity in our relevant tissue. After that, we make predictions at the orthologous or corresponding regions in a large number of species. We then associate our predictions with brain size, and then we evaluate the statistical significance of our association. We call this method the Tissue-Aware Conservation Inference Toolkit, or TACIT. I'm now going to explain in a little more detail how I implemented each of these steps to study the evolution of brain size residual, beginning with the first step of TACIT. My lab mates Morgan Worthland, Alyssa Lawler, and Ashley Brown generated open chromatin data from 2 tissues, motor cortex, a region of the brain, and liver, in mammals of different levels of genetic relatedness, and we supplemented this with some publicly available data. And for the remainder of this talk, I'm going to use open chromatin regions as a proxy for enhancers and closed chromatin regions as a proxy for non-enhancers. But that said, a major caveat is that there are many open chromatin regions that do not actually function as enhancers, and open chromatin assays usually fail to miss many enhancers. For the remainder of this talk, I'm going to be focusing on our results from the motor cortex, because that is the tissue most relevant to brain size, and I'm happy to tell you about our liver results offline. So now that I've told you about how we generated data, I'm going to tell you about how we trained our machine learning models. So what we have is we give our model sequences from the species for which we have data, and we label these sequences as either a 1, meaning this sequence underlies an enhancer in our tissue, or a 0, meaning that it underlies a non-enhancer. More specifically, our positive set, which are our 1s, we used our open chromatin regions. And for our negative set, we used what I call non-enhancer orthologs of enhancers. These are closed chromatin regions whose orthologs or corresponding regions in another species for which we have data are open. And I chose to use these regions. This forces the model to learn what differences between species lead to differences in enhancer activity. And then at prediction time, we can do is take a new sequence for our model and ask the model, is the sequence an open chromatin region in our tissue? The type of model that we decided to use was a convolutional neural network. So I actually experimented with multiple different negative sets, and additionally created a new set of machine learning model evaluation criteria designed specifically for this task. Since time is limited, I'm not going to be able to go into those results, but you can learn more about them in my paper that was published in BMC Genomics. So here are the results from our models. What I'm showing you here is the AUC and AUPRC. Both of these range from 0 to 1, with 0 being the worst performance and 1 being the best performance. What we'd expect at random for AUC is a performance of 0.5, and what we expect at random for AUPRC is given in the white bars. And these are the bars, different colored bars, are from different evaluation criteria that I developed. And what you can see is that our motor cortex models had extremely good performance across evaluation criteria. So now that I've told you about how we trained our machine learning models, I'm going to tell you about how we made predictions at orthologs in different species. More specifically, what we wanted to do is what we have is a matrix that looks like the one on the top, where each row is a candidate enhancer, each column is a species, and we have data from a small number of our species and most of our data is missing. What we want to do is get the corresponding regions or orthologs, and then use our models to make predictions at them, allowing us to fill in most of this matrix. And the way that we decided to obtain the orthologs is to use the alignment from the Zoonomia Consortium of over 200 mammalian genomes. And specifically, we focused on the Boreoeutherians, which are the mammals in the black boxes. And our reason for using only the Boreoeutherians is that we were not able to obtain open chromatin data from more distantly related species, and therefore have no way of knowing whether our models can make accurate predictions in those species. And the specific process of getting the orthologs was done using a combination of tools published by others in bioinformatics and published by me and a fantastic undergraduate of mine in bioinformatics. So now that we have our candidate enhancers, our orthologs, and our predictions across large numbers of mammals, we needed to find a way to associate these predictions with phenotypes. And to do that, I'm going to describe how we implemented the fourth step of TACIT. To motivate the way we did this, I'm going to present a toy example of a candidate enhancer in the rhesus macaque. In this toy example, we have our predictions at orthologs and a number of other mammals and our phenotypes. And what you can see is that there is a strong positive correlation between our predicted enhancer activity and our phenotype, suggesting that this enhancer should be a high priority for follow-up studies. However, if you look a little more closely, what you can also see is that there's a strong negative correlation between our predicted enhancer activity and how far our species is from the rhesus macaque, which means that it is impossible to know if our enhancer is actually involved in our phenotype or if this entire result is due to the confounding factor of the phylogenetic tree. We therefore decided to use a method called phylolome, which is a sped up version of phylogenetic generalized least squares. And the high-level idea of how this works is that we use a generalized least squares regression to find the significance of our association between predicted enhancer activity and our phenotype, but we have an expected variance covariance matrix that's based on the phylogenetic relationships between species so that the associations we find cannot be entirely explained by phylogeny. And for binary phenotypes, we use the logistic version of phylolome called phyloglm. So now that I've told you how the fourth step of TASA works, I'm going to describe how we evaluated our statistical significance of our associations. To do this, we used a method called phylogenetic permutations, which is a type of permutation test where we permute our phenotypes in a way that accounts for the phylogenetic tree structure. So I asked a fantastic undergraduate of mine, Daniel Schaefer, to run phylogenetic permutations for approximately 100,000 motor cortex enhancers and for a number of different phenotypes. And he came back to me and said, this method is way too slow, especially for binary phenotypes. He then decided to come up with a speed up for this method that decreased the runtime by a huge amount, decreasing it by over an order of magnitude for binary phenotypes. You can learn more about this speed up using in our paper, and you can check it out at our GitHub. And I'm excited to say that Daniel has decided to continue his scientific career and has recently started his PhD at MIT. So using TACIT, we identified 49 motor cortex enhancers associated with the evolution of brain size residual. And Daniel and I then did a massive literature review on the genes near these enhancers to see if they might be previously implicated in anything related to brain size. We found that 14 are near genes associated with microcephaly or macrocephaly. In fact, enhancers near genes associated with micro or macrocephaly tend to have stronger associations with brain size than other enhancers. In addition, 17 of the other enhancers are near genes previously associated with other neurodevelopmental disorders, and 8 are near genes implicated in the growth or spread of brain cancer. So here we have an example of our predictions for one of the candidate enhancers. On the x-axis, I have our predicted motor cortex enhancer activity. On the y-axis, I have the brain size residual. And for the purpose of not making this plot too crowded, I've just plotted points for the cetaceans, which are the whales, which have the largest variation across any mammalian clade in brain size residual. But as I mentioned before, no one previously has figured out the mechanism underlying this variation. So here what you can see is a strong negative association between predicted motor cortex enhancer activity and brain size residual. So then here we can zoom in a little more on our predictions and our phenotype annotations, and we can look specifically at the bottlenose dolphin, which has a large brain size residual, and our closest species with a genome and zoonomia, the killer whale, which has a much smaller brain size residual. And you can see here a very large difference in predicted enhancer activity in the opposite direction. Here we have 2 more whales with a large difference in brain size residual, and once again we see a large difference in predicted enhancer activity in the opposite direction. Here we have 2 mink whales that both have large negative brain size residuals, which means that their brain size is much smaller than we'd expect based on their huge body size. And one is a more negative brain size residual than the other. And we see once again for these mink whales, a difference in predicted enhancer activity in the opposite direction. So we then ask this a question, where is this enhancer in the genome? Here you can see a browser shot from the common mink whale, and our enhancer is indicated in the red box, and you can see that it is near the gene LRIG1. So then I wanted to know, does this enhancer actually in 3 dimensions interact with the start site of LRIG1? So Daniel reprocessed a three-dimensional interaction data set from mouse, and we decided to use mouse because mice have small brains. And what he found is that the enhancer ortholog has a significant physical interaction with the LRIG1 transcription start site. LRIG1 is expressed in the mouse cerebral cortex, and LRIG1 regulates quiescence in adult neurostem cells and neurostem cell proliferation during development. Here's another example of a candidate enhancer. You can see here this browser shot in chimpanzee, and this candidate enhancer is negatively associated with brain size residual in the great apes, but it's actually not the enhancer identified in the previous study because it does have a human ortholog. The human ortholog is just predicted to not be a motor cortex enhancer. So the nearby genes are TAF1D, MED17, and VSTM5, and all three of them are expressed in the mouse cerebral cortex. Mutations in MED17 are associated with microcephaly and other neurodevelopmental disorders, and VSTM5 regulates neuronal morphology and migration. So I then asked the question, why is it that this candidate enhancer's ortholog is so confidently predicted to be an enhancer in the chimpanzee, but so confidently predicted to not be an enhancer in humans? To try to understand this, I interpreted what transcription factor sequence patterns had been learned by our models, and then scanned the chimpanzee ortholog for those patterns. And here's an example of what I found. This sequence is probably familiar to some of you because it matches the motif for the transcription factor CTCF. Now, if we look at the human ortholog, what we can see is that this G in the CTCF motif is an A in the human ortholog. This suggests that the human ortholog may not be a candidate enhancer because it cannot be bound by CTCF. In the future, I want to extend this work to investigate the evolution of metabolic phenotypes. And I'm going to start off by focusing on adipose and liver tissue. And some specific phenotypes I'm excited about are dietary phenotypes like carnivory and herbivory and hibernation phenotypes. And if you are interested in doing this with me, I would love to recruit you to my lab that I will be starting at Carnegie Mellon University next summer. I'm recruiting people at the PhD student and postdoc level, both computational biologists and experimental biologists, as well as people who started out in one area and are interested in transitioning into another. In conclusion, we have developed Tacit, which associates predicted enhancer activity of candidate enhancer orthologs with phenotypes. We use Tacit to identify motor cortex enhancers associated with brain size residual, and Tacit can be applied to any phenotype with annotations in dozens of species and enhancer activity data in a relevant tissue or cell type from at least a few species. And you can learn more about this work and about the many results I didn't have time to go over in our paper that recently came out in Science. With that, I would like to thank my postdoc advisor, Andreas Spenning, everyone in the lab who has in some way contributed to this project, my funding and compute resources, and my many collaborators, and I would be happy to take any questions.
Thank you so much, Irene. That was fantastic. I'm sure there will be quite a few questions. Someone actually already had a question earlier in the seminar where Stephen Kravitz asks, what happens in the case of super enhancers, which is interesting, I think.
That is a great question. So we made the tough decision to actually eliminate anything that we thought was probably a super enhancer. So we took open chromatin regions more than a kilobase and just didn't use them. What I found is after I trained my models, I tried doing predictions in these longer open chromatin regions. And what I found was that as the length increased, the model performance went down. I think that's because for shorter open chromatin regions, you have a peak summit around which a lot of the motifs are centered. But for these longer ones, there might be multiple clusters of transcription factor binding motifs. So the model may need to learn something different. I think extending this work to super enhancers could be really exciting. I'd have to see if there's enough to train a model. You might need a different kind of model. We may want to pre-train on existing, using some kinds of existing enhancer models and then fine-tune with super enhancers or something like that. That is something I would love to do in the future.
Yeah, maybe add some K27 acetyl data or something.
Yes.
There's another question. Great work, Irene. I agree. Have you also potentially looked at transcription factor motifs on 49 enhancers? And if you have, do they also indicate involvement in brain size, etc.?
Okay, good question. So the CTCF motif that I showed you was actually one of the few that I found. A lot of the time when I used a method called deep lift for interpreting the models, and then I used a method called TF Medisco for summarizing the results. A lot of the time when I looked at the differences in enhancer activity, places where we had different enhancer activity between species and looked at the deep lift scores, there were sequence patterns like coming up as having been found by deep lift. They did not match known transcription factor motifs. And sometimes they weren't even in the summary from TF Medisco. So I think that there's a lot more that needs to be done, both in terms of interpreting these results and in general understanding what sequence patterns lead to enhancer activity, as well as a lot more work that needs to be done in coming up with ways of summarizing these nucleotide level importance scores from convolutional neural network model interpretations. And I know some current members of Anshul Kundaje's lab, where I did my PhD at Stanford, are working actively on these questions.
I have a question too, but I just also wanted to point out, if anyone wants to ask a question by voice, just remember you can raise your hand and ask that, and I can invite you to unmute yourself. Okay, so yeah, I was excited about the LRIG-1 result because I knew that it was involved in quiescence regulation. I was wondering if how commonly found genes were involved in quiescence regulation in this, if you remember off the top of your head. You probably don't because you have so much info.
A large number of nearby genes were somehow involved in quiescence regulation.
Yeah,
I wanted to do some kind of statistical analysis to figure out, like, is this more than expected by chance? Is there some kind of pattern? We were able to do that for micro and macrocephaly because OMIM has annotations. So Daniel wrote a script to scrape OMIM and get that information. There isn't, to my knowledge, a quiescence regulation database specifically for the brain. If you know of one, I'd love to know what it is.
Yeah, we don't have one yet, but
we didn't know of a good way to scrape PubMed for this information, so we weren't able to do that. So yes, we definitely found a lot, but I don't want to emphasize it too much because I don't actually know if it's more than expected by chance.
Yeah, this is one of my like selfish questions that I think a lot of people probably roll their eyes at when people ask, but I had to ask because I was interested.
No, it's a great question, and I had it too.
But I also, can you remind me, ironically, my brain's a little off center today. So can you remind me, what tissue of the brain was, or what part of the brain was used for the?
Thank you. We used the motor cortex, and what I didn't have the chance to go into is we didn't have any reason to think the motor cortex would be more involved in brain size than other regions. One of the other phenotypes we worked on was vocal learning. That paper is currently under revision. We have a lot of really interesting results, and that was based on work done by my postdoc advisor when he was a PhD student, demonstrating the involvement of the motor cortex gene expression in vocal learning evolution. And I should say that actually using consistent brain regions across species is really important for being able to train machine learning models. We found that our models trained in motor cortex did not give as good results in other brain regions. And actually, one of my criteria for evaluation that I developed was, can it predict open chromatin differences between brain regions, which we found it actually worked okay on.
Okay, cool. I was curious because I thought you said motor cortex, but I wanted to double check. How close is the motor cortex across species to like hippocampus? Because that's where a lot of the quiescent neural stem cells are.
Question. I don't think it's particularly close.
Okay, interesting.
My intuition, and I have some ideas for how to actually see if I'm right, is that a lot of these enhancers open up earlier in development before all the brain regions have specifically formed, and then just stay open, which is why we're able to find them in adult tissue. My postdoc advisor was actually queasy about us working on brain size, but Daniel and I insisted because there was so much phenotype data, way more than we had for a lot of other phenotypes we thought were more promising, that we felt like we just had to try it.
Well, I have more questions, but I can't keep hogging this, so I'm gonna let Shubham, would you like to unmute yourself? Or yeah, you have your hand raised. Yeah, there you go.
Thanks for the great talk. So I think at some point, unless I am misunderstood, you mentioned that you couldn't look at some species because you didn't have the open chromatin information for them. I have a two-part question. So first is, how are you defining the open chromatin region? Like, is it based on ataxic or like histone modifications? And second, since you are looking at enhancers, have you thought about considering using Hi-C maps as the input instead of the open chromatin annotation itself?
Yeah, those are great questions. So yeah, good question about open chromatin. For this study, we used ataxi. We've also trained models with DNase hypersensitivity and found that they worked pretty well. Yeah, we didn't use, when I said there were species we couldn't use, what we do is if we have a species with data in our clade of interest, we'll make predictions at the orthologs and all the other species in the clade on the tree. But if there's a part of the tree where we have no data from any species, we don't make predictions there because the models assume that there's this conservation regulatory code and we can't actually evaluate that. So I would love to use Hi-C data as input to these models. The reason I chose not to is that we didn't have really high quality Hi-C from multiple species in the same part of the same tissue. So that's the kind of data we need. I've actually been talking to a research scientist at Duke, another research scientist at Duke who knows someone who might be generating that data. So I'm hoping that's something that we'll be able to do in the future. And I think probably some differences in types of machine learning model setup are going to be needed to actually incorporate the 3D interactions and things like that. And I know there's some people already working on machine learning models for predicting Hi-C loops. So I'm excited to move in that direction.
Thanks very much.
Awesome. Thank you so much. I think we're at time, so we should go ahead and give the stage to the next speaker. But thank you so much, Irene. That was fabulous.
Thank you.